In front of us, we are having a 3D structure of protein with disulfide bond. Now, disulfide bonds are very important for the stability of many proteins. They form when two sulfahydryl groups of cysteine amino acids come into a right angle, and then the process of oxidation will occur, hydrogen will be removed, and there will be formation of a disulfide SS bond. Now, there are many interesting things about disulfide bond, and one of the important thing which I find interesting, that is which are the proteins which are having disulfide bond and which are the group of proteins which don't have disulfide bond. Giving answer is very simple. The environment of our cells is reducing in nature. It means if a protein or a polypeptide which is containing disulfide bond will not be able to retain it in our cellular environment, because the reducing environment will do the reduction of SS bond and will convert back into SHSH. So, if a protein which is present with disulfide bond in our cytoplasm will not be able to retain this bond. Now, if a protein which is packed in vesicles containing disulfide bond and transported to the plasma membrane or transported outside of the cell will be containing disulfide bond, because this protein is protected from the reducing environment of the cell. So, I can divide all the proteins into two major classes, one which are remain in the cytoplasm and don't contain the disulfide bond because of reducing environment, and the second class of protein which are present on the plasma membrane or secreted out, they can have disulfide bond because they were packed into the vesicle and they were sent to the plasma membrane or outside of the cells while protecting the disulfide bond from the reducing environment of the cytoplasm. Now, I am having one very interesting analogy to explain this. Imagine cell as a country. It is having two types of people. One, civilian people which are working inside the country, and our proteins which are present in cytoplasm, they can be considered as civilian protein because they are working in the cytoplasm. Now, civilian proteins don't require extra stability. So, you don't need disulfide bonds, so you can have them into the reducing environment of the cytoplasm. So, the proteins which are present in cytoplasm, they are civilian proteins not requiring extra stability, so there is no need to have disulfide bond within them. Now, the proteins which are working in the plasma membrane are secreted outside, like insulin or antibodies or some receptors on the plasma membrane. They are working on the borders or they are working outside of the country. So, we will call them as soldier proteins, and soldier proteins, they are going out of the world, out of the country. They require extra stability. For extra stability, they should have disulfide bond. So, what do you do with soldier proteins, or what do you do with the soldiers in the country? You train them in a specific compartment, let's say endoplasmic reticulum. After training, you send them to the border or outside of the country in the special military vehicles, and those special military vehicles in our cells are vesicles. So, civilian proteins remain in the cytoplasm, don't require disulfide bond. Military proteins present on the plasma membrane or working outside of the cells, they require extra stability and they have disulfide bonds. One very interesting case is of archaea. You can imagine archaea as a country which is continuously in war, because they are continuously in stress or in the extreme environment. So, they should be having most of the proteins as soldier proteins to tolerate this continuous war or continuous stress. So, archaeal system is having a microenvironment within the cells which is allowing the disulfide bond retention in all the proteins. Means in the archaea, most of the proteins are requiring extra stability and they are having presence of disulfide bond. With this analogy, we are able to say which proteins are requiring disulfide bond or which proteins are not having disulfide bonds.